Welcome, everyone. It's my pleasure to have again Frank David with us. And for those that don't know who he is, he's the founder of Pharmagellan. I think we see him also as a US biotech guru. And he's also the author of the book Analyzing Biotech Clinical Trials, which is a one-of-its-kind book. I think we recommend it for to all investors that want to invest in the space, because it really puts all the important pieces together. We will go through 3 cases today: BioArctic, Vicore Pharma, and BioInvent. BioArctic is about to have a phase 3 readout in Alzheimer's in September, one of the few companies still in the race at this stage. And Vicore Pharma is engaged in fibrotic diseases and had a very interesting early data in February in idiopathic pulmonary fibrosis, very tough to treat rare disease. And then we will go also to BioInvent, an immuno-oncology company that tries to navigate through the jungle of targeted new treatments where it's really difficult to interpret. And hopefully Frank will be able to clear a few things up for us. Before we do that, I think it's always interesting to do a bit of a sector recap and outlook. Just this week, there was another bigger event with the TIGIT blow up, if one could say so, from Roche, that also affected other companies. Yeah, really excited to hear your view about the sector.
Yeah, no, thanks for having me. Pleasure to be back. I think there were maybe two or maybe 3 interesting things to say about what's going on now. One is sort of, even backing away from TIGIT for a second, obviously there's a lot of chatter, I think, in US biotech, certainly, that valuations are so low that this is going to sort of trigger a big buying spree among large companies. I'm not so sure that that's true. I think it is true that valuations are low and large companies are stacked with a lot of cash. But at the same time, assets that are worth buying are worth buying, and assets that aren't worth buying are not worth buying at pretty much any price. And I think that just gets to sort of understanding the mindset of larger companies when it comes to doing these types of deals for R&D stage assets, which is twofold. Number one, every large company recognizes that taking one of these things on, there's still a tremendous amount of risk, and they have to decide whether they're going to take on that risk. Number 2 is it also just involves a lot of human capital, right? So, often the thing that's limiting in these large companies is there's not unlimited human capital ability to run as global project heads and people to really shepherd these projects. Yeah. So really deciding to take on one of these projects, even if there's technically enough money to take on that project in addition to the pipeline, often there's just not enough qualified people to be able to run that side by side with the pipeline, which means that if Merck or Pfizer or whoever takes on one of these R&D stage projects, it means that they have to decide to deprioritize something else that's already in their pipeline. And I'm not sure that that's always so attractive when those things get lined up, especially since there's an information asymmetry. The things that are in your pipeline, a lot better, which means you're sure the problems, but you also know the good things. Whereas the things that you're looking at from the outside, often there's just a lot of unknowns and you don't know them as well. And so they end up sometimes looking worse. And then, so maybe I'll just stop there in terms of sort of that part of the puzzle. I think in terms of TIGIT specifically, this is an asset which I think followed what, we'll talk about it a little bit later, but, followed kind of a classic playbook of having sort of very mediocre monotherapy activity, but then having a glimpse of activity as combination, and then use that to move forward. Obviously a very hot target where there was a lot of good scientific reason to be excited about it. And I think, there's been a now multi-year hunt for these types of things that could synergize with and boost the effectiveness of the immuno-onc agents. But at the end of the day, it looks a little bit more like IDO, which for people who don't remember that story, essentially looked the same: mediocre to negligible monotherapy activity, looked promising as combination, and then just didn't pan out in actual RCT. So in many ways, this is a song I think that we've heard before. And although it's always unfortunate when these things happen, I think anyone who goes into one of these combination types of situations without monotherapy activity, I think recognizes that that's a risk. I'm not sure that there's anything specific about TIGIT that would make it more or less risky in that perspective than other things. And then the third thing that I would just mention again, which maybe goes back to the first point, is people have been bemoaning sort of why isn't there more M&A going on in the sector? And then we had Pfizer buying Biohaven for many billions of dollars. And I think it's interesting to kind of remember for us all to be humble and remember that when given the choice between buying a dozen or two pre-commercial biotechs versus buying one revenue-generating company, Pfizer decided to buy itself some revenue, and specifically, basically buy something that's de-risked and where it's obvious where the synergies come from. That's a situation where it's worth more in Pfizer's hands with its commercial capabilities than it is in Biohaven's hands. And I think that should probably give people pause in terms of getting all excited about the amount of firepower that's on the balance sheets of a lot of big pharma companies and thinking that that's going to unleash this wave of M&A. I'm sure there will be some acquisitions of pre-commercial biotechs, but I think it would be a mistake to think that there's going to be this deluge of deals because valuations are low and cash balances are high.
Thank you. That's a very good overview. So yeah, it sums it up. So the risk stays in this sector, no matter what data you have at the earlier stages, and you don't know how the partnerships will be. And as you said a bit earlier, that in the end the good ones will get the deals, the bad ones won't. And for the bad ones, no matter the low valuation, it's still too high.
Exactly.
So we'll move into the first company case, that is BioArctic. So I think many people are now looking forward to it, both as an investment and also from the clinical side and from the hope of having finally one type of treatment that could slow the progression of Alzheimer's. So what's your view on the phase 3 design and the data that goes into the phase 3, the imbalances we saw in the phase 2 and the effects of that on the trial?
Yeah, I mean, I looked at it. I think there are a couple comparisons worth making. Obviously, you can look at the pre-existing data, the phase 2b data, compared to what they're planning for phase 3. You could also look at it compared to the Lecanemab phase 3, right? And then you can compare those two phase 3s against each other, right? So that's sort of the context. That's, I think, the context for how to look at it. I mean, the good things about the existing data from BioArctic is, the drug seems to have a better safety profile than Lecanemab did, in terms of the ARIA incident. And even though it was a statistical fail, on technical terms, clearly there's some evidence of dose responsive effect if you look at the clinical scores. And just to be clear, I don't really look very hard at the sort of effects on tau and other types of things like that. I mean, to me, this is all about really the effects on the clinical endpoints. Whether you're talking about the CDR endpoint or whether you're talking about or ADCOMS or whatever, the thing on the y-axis needs to be clinical, I think, to really get excited. And I think even more so now in this field. So it's a drug that seems to maybe be doing something and seems to be doing it maybe safer than Lecanemab was. So that's good. I think the phase 3, it's hard. I didn't do sort of any sort of attempt at a formal power calculation, but much larger, obviously, than the sample size that they tested in phase 2. And in phase 2, they were nominally looking for a 25% improvement in where the scores would end up. So, it seems on its face like this new trial, the phase 3, is probably powered to detect a smaller difference than that.
Yeah.
Now, of course, that's glass half full or glass half empty, depending on how you look at it, right? On the one hand, it might, it probably means that there's a higher quote unquote probability of success for the phase 3 study than one might have otherwise predicted, because they're sort of saying, well, now maybe if we hit 12 or 15 or 18% improvement, maybe that'll score as a win. On the other hand, will that matter to regulators or to clinicians, I think is the bigger question. And I don't think we really know. Again, this field is so nascent in terms of really understanding what people will think. You go to physicians and you start asking them in a field like this, well, how big a difference would you have to see to be meaningful? And you're talking about something that's a little bit, how can I say, it's not vague, but it's hard clinically to kind of put your finger on what does it mean a 25% less rapid progression, right? 25% less progression over 18 months. What does that actually mean clinically? People don't really have a good grasp. It's hard to wrap your brain around that, right? It's not like one of these things where you're saying sort of length of life, or where you're talking about sort of some clear functional scale where you weren't able to do something and now you're able to do it, or pain or something like that. So I think it's just hard to know how important that that difference will be seen to be. And I think the safety question is again, it's good, better than having worse or equivalent amount of ARIA. Yeah. How will that influence anyone? Who the heck knows? I think that's kind of an open question. As you mentioned, I mean, they had a situation with the ApoE4 where they had to basically, because those are the patients who seem most likely to respond, but they had to back off of giving them the highest dose because they were also more likely to get the ARIA side effect. So what does that mean in terms of how this study shakes down and in terms of the eventual label? I think it's a great question, and I don't really. I think that will play out in real time. I don't have anything particularly insightful to say there, but that's certainly something I'd be watching for as well.
But very interesting analysis. So at least on the kind of on paper, what they could do right, they seem to have done right. So they have enough patients in there. They have fairly good readout from the phase 2. The safety looks good. Now the question will be if the data is very weak on the efficacy, if it could get approved like Aduhelm, but then it could face the same issues as Aduhelm and not generate any revenues. So it's probably then, so there's still the question is if it's positive at all, of course, but at least they did everything right. Or
well, yeah, I mean, I think there's two issues there, in terms of doing everything right to me. One is what we're likely to see if I were a betting man, I'd be betting on sort of a mixed picture on right. Their primary endpoint on the phase 3 is the CDR scale, which is not the same as what their main endpoint was on in the phase 2. The CDR, my understanding is that that's viewed as sort of a more stringent. It's a tougher bar than some of the other scales. Yeah. So it's quite possible that they could not hit on that, or maybe they would hit on that because they have a bigger sample, but the effect would be less impressive than it might be on some of these other scales. So what are you going to make of that? What are the various stakeholders going to make of that if there's kind of an inconsistency in terms of the effect size? I guess that's one piece. And the other piece is just kind of the double-edged sword of the sample size, right? Where there is always a risk with these big studies that you end up winning the battle but losing the war, right? That you end up getting a positive result, but then people say, well, let's just take the ridiculous case. Let's say that it's so overpowered that they could detect a 2% improvement over baseline. I don't think it is that overpowered, but let's just say, for the sake of argument, you could then imagine a situation where you say, well, 2% just isn't that much. And meanwhile, there's still a bunch of ARIA, this is really not approvable, or even if it's approvable, we're not going to buy it. We're not going to pay for it. So where is that? You know, somewhere between 2% and 25% is probably the threshold where it becomes not as interesting, you know, to regulators or to clinicians or to payers. And I don't know if any of us knows exactly where that line is, right?
Okay.
So I think we're only going to know after we see where their data shake down on the phase 3. But I think that is the risk that you're buying into, which is, you know, it's unlikely that the difference is as high as 25%, right? Because that was how their phase 2 was powered. You got to, if you believe in sort of reversion to the mean at all, you would figure that the phase 3 is likely to be maybe a little less impressive than the phase 2. So let's say that they could have shown a 20%, you know, improvement in the phase 2, and let's say that now they show 15. And again, we'll just have to see what people think of that. You know, it may be that's a little bit more regulatory inside baseball than I understand, honestly. You know, trying to figure out whether
it raises important questions now to the upcoming readout. So it could be positive from a clinical readout perspective, but then we would probably want to see around 20% difference to feel comfortable that this is really will be a value generating asset, a revenue generating asset.
Absolutely.
Below, it becomes more and more tougher to see that, but could still be above. If they have 30%, the chance is very high.
Yeah, right. I think that, you know, I would say I wouldn't, and I'm not an investor, but if I were an investor, I don't think I'd be investing in this thinking that they're going to knock it out of the park, thinking that there's a sizable chance that they're going to knock it out of the park, just given what we've seen in the phase 2 and given the comparison with Lecanemab and given what we know about the mechanism. So, you know, you got to sort of, the chance that this is going to be a home run, I would say, is low. The more likely scenario is that there is a statistically significant effect, and we're all left thinking of trying to figure out like how clinically meaningful that is. I mean, I will just add that every time I've done this, you know, go out to doctors and say, well, what difference would be meaningful to you? It's a little bit of a hand-waving argument. You know, doctors are bad with numbers, and nobody can tell, you know, people will tell you, oh, they'll give you some round number, like, oh, 25% difference, 20% difference, whatever. But at the end of the day, if you then give them a drug that they can prescribe to patients that gives a 15% improvement, many of them will still prescribe the drug because the alternative is to not prescribe the drug, right? And to not take that 15% improvement. So, you know, I think that, and that's where I think having the better risk profile, I think is, so I think from an opportunity and valuation perspective, it becomes challenging just trying to understand how those things fit together. You know, how does the clinical performance of the drug match up with really what the size of the opportunity is? But you got to believe that even at 15%, you know, patients are going to be walking into their doctor's office asking for it if it gets approved and is going to get paid for. So, you know, so that the wind's at their backs in that sense. They probably did the right thing by running quite a large trial.
Especially as you mentioned, that the data will come from a proper trial. It will not be some special analysis such as Adrielm had, and the safety profile is better. So there is nothing else on the market. So there could still be a lower bar than.
No question. No question.
I think you also mentioned the scales. I think something also you mentioned in the book on the rheumatoid arthritis, osteoarthritis, and schizophrenia. How one interesting example, I think, was where you mentioned that there was in one case a smaller scale for the same disease and then a bigger scale, and on so you have the different readout. On one it is not significant, on the other it is significant, but both are measuring the same, just having a bigger scale. So one was from 0 to 40, the other from 0 to 60 or so. So how can we look at the scales here?
It's hard to know. These are complicated problems. I mean, I did look up a little bit about the you know, the ADCOM scale just before we got on the call because I was not familiar with it, honestly. You know, ADCOMS is basically a composite that takes the CDR scale plus the mini-mental status plus some components of ADAS-Cog and sort of matches them all together, essentially. So, you know, there's been some amount of kind of, you know, research and thinking about it, you know, since it was originally, it's relatively new, you know, since it was originally proposed as a scale. again, this turns into a question of sort of what's the perceived clinical value of a given level of improvement on that scale, right? So, and I think that those are the sorts of things where we don't have, in an area like Alzheimer's, where we don't have a bunch of other drugs that we can then run through the gamut and analyze with the same scale and say, okay, well, drug X gives us this amount of improvement. So now let's sort, and we like drug X already and we have experience with it. So now we can sort of say, okay, like now with this new drug, now we understand what a 20% or a 15% improvement is on this scale. So I think that was sort of that's certainly, one of the challenges of using these new scales like the ADCOM scale. The phase 3 is powered for the CDR scale, which I think has a longer history behind it. So from a clinical point of view, probably will resonate more with regulators and physicians probably will understand that better. But you're always, with these, there's always a complexity in terms of sort of thinking about what's the actual clinical meaning of these, right? Because they're all taking various, whether it's physiologic measurements or whether it's things from metastatic, physician elicited kinds of questions or biology or what have you. They're all kind of mixing them up in different ways. And there's not a right answer, except I would say that in a field like Alzheimer's, again, because we don't really have approved drugs, on the one hand, CDR is clearly is a scale that everyone understands, and ADAS-Cog is one that everyone understands. I'm not sure why they chose to power theirs for CDR. I'm sure they had some good reasons. And if I were trying to imagine why they did, it would probably be because they felt like it was sort of the one where getting a positive, as long as they're going to invest in overpowering the trial anyway, maybe they figured, look, this is perceived as the most stringent scale. So therefore, if we win on this, nobody's going to mess around with us. Whereas if we win on something like I keep having to look at what the name is, like AdComs, if we win on something like AdComs, and then maybe we don't win on CDR, then we're going to raise all these questions about, well, what do we really do? So I could imagine that might have gone into the thinking. Like, look, if we're going to overpower anyway, let's overpower on a scale that we're pretty sure nobody is going to question us about.
Okay, very insightful, and I think some nice conclusions we can take despite the remaining risks. So a well-designed trial, it's overpowered, not underpowered,
almost certainly. Yeah,
a good phase 2 base, a good side effect profile. There's no approved treatment, so a lot of positive things. But of course, we need to remember it's Alzheimer's. It's a very tough nut to crack, and we look excited to September and on that one. Now to maybe a more clear one, if one could say so, Vicore Pharma.
Sure.
So they had interim readout in idiopathic pulmonary fibrosis in February, where they showed unprecedented increases in forced vital capacity, the lung volume measurement that is done usually in these trials. And my first question was, something wrong with the numbers, because these kind of increases in the hundreds of milliliters, we have not seen that from the other companies that some of them recently failed, like Galapagos. That's maybe a question. It's the small numbers. Can that be a coincidence, or how do we take that data? Do we even need to conclude that this is now a home run, or can we just see this as exploratory? Or
a great question. I mean, the phase 2. So first of all, you just look at the picture in the phase 2, and it's crazy. I mean, as you pointed out, you've never seen anything like this, right? Where the quote unquote control is going down and the drug treated is basically staying or maybe even improving. Is just crazy pants. I think then you dig into it a little bit more, and that's where you start to sort of recognize that maybe, it's still certainly promising, but maybe with a little bit of caution thrown in there. It was a single arm trial, so the control arm is actually kind of a synthetic. It's basically derived from the literature. It's not an actual true RCT controlled study. So, I would say that ends up being a big issue. There is a lot of There's a lot of interest at the FDA in using these so-called synthetic controls or external controls for studies, but the bar that they put here is really just kind of coarsely derived from other literature-based results from the placebo arms of trials of other approved drugs. That's not really what FDA means when they talk about kind of an external control arm. When FDA talks about an external control arm, they really are talking about a situation in which you try to match the patients on key characteristics between what you had in your trial and then some other external source of data. And that's not what we have here. So, what you worry about, obviously, is a scenario in which the patients who were treated who were on drug in this study actually were somehow healthier or less likely to progress, let earlier stage of disease than the ones that you're quote unquote comparing against, the ones who participated in the phase 3 trials of some of these other drugs. And, that would imply that maybe if you would have actually run a true RCT, that both groups would have just been basically, there would have been very little difference between those lines. Again, you'd be much more worried about that if their drug had gone down too, and you were comparing sort of like in the Alzheimer's example we just talked about, everybody's decreasing, but you're just talking about who's decreasing 25% less. That would make you very concerned in this case, because then you would say that, well, I could clearly imagine that a pretty small difference in the population could account for that, like slightly healthier patients. Here I would say, again, it's impressive that the patients really over that period of time did not really decline and might have actually improved a little bit. So, certainly, again, intriguing, especially in an area like this. But keeping in mind, the upcoming phase 2b is going to be the first real RCT look at the effect size. And, that's really what you need in order to sort of power a pivotal study and understand really what the magnitude of the effect is. So
that's the next challenge for the company. So
absolutely. And that's really, that's where I'd be looking toward, is sort of as the company, it seemed like they still have not decided even what the control arm is there. And, there you start to think about, okay, what types of patients are they going to go for? Are we going to primarily be looking at earlier stage or later stage patients? How are we going to tell that based on the baseline characteristics of the patients? And how are the inclusion and exclusion criteria going to be set up to either favor one of those groups or another? And then, then we'll get a real look at how it does either versus placebo or versus one of the standard of care agents.
Yeah. So we can sum up a bit here that the data at least doesn't look bad. So safety they also got from the COVID trials.
Yeah, no question.
That looks good, but it's an open label trial that was not controlled, so single arm, and but so far it looks better than what we have ever seen in IPF. How do you inform a randomized clinical trial here? Also, in terms of powering, you can't if you assume these kind of increases, you would underpower your trial probably.
I think that's why, in a phase 2b, I don't think you'd go to a registration trial, and I don't think they're intending to go to a registration trial from what I can gather. I think they're going to do a phase 2b type of study where, it's going to be, again, somewhat exploratory, and there's a chance that trial loses the battle but wins the war, right? Like it could be, to a certain extent, that phase 2b could still be somewhat exploratory, but if it gave you good information in order to inform a phase 3, right, it kind of gave, because now, regardless of whether that trial, sort of like the example we just talked about, right, in Alzheimer's, right, it almost didn't matter that much whether the earlier trial did or did not meet its pre-specified endpoint, because it gave enough information to design a more likely to succeed trial. So I think the same is true here, like getting a better glimpse in a real RCT setting where you've set up real inclusion and exclusion criteria of what effect size to expect, regardless of what assuming there's any difference there, which, one would hope there would be, will provide information to be able to inform a pivotal trial. So I'm not sure I'd be looking at the next study. I mean, obviously there's a chance that the next study would totally be a null, that there'd be no difference. Anything is possible. Again, I would say that given the, I would say that's less likely to me, just given that, given how good the drug-treated arm did in the single-arm study. So I don't know that I'd be viewing the upcoming phase 2b trial as necessarily a sort of go, no-go catalyst, for either the company or for investors. It's mainly just to generate additional information to inform the pivotal trial.
Yeah, I think the company has expressed the potential that it could be a pivotal. I guess that is in a blue sky scenario that they have a randomized trial against standard of care and beat it by far with such data. Then probably the FDA and the EMA would be willing to, based on also the side effect profile, if it is similar with that mechanism, I think, which is interesting. It's an agonist on something that is overexpressed only in the fibrotic tissue and placenta. It's not blocking something like many of the other drugs. So I think that is and I try to put all the pieces together, but from the preclinical data where we have data from in the same models as the approved drugs, the long COVID data where we can see there is an effect on the lung. And now this data, it all is good, but yeah, it's probably one of these cases where one could say, from what we see now, it would justify further investment. It's
well, I mean, I think this is almost the inverse possibility, the inverse of the Alzheimer's example, right? The Alzheimer's example, we were just saying that the odds of a home run are very, very low, right? We're either it's going to be pretty mediocre, or it'll, a mediocre success, or it'll fail, right? Yeah. Whereas here, I would say, again, there's certainly a chance that they produce data out of the phase 2b that are comparable, that are roughly comparable to the data that from their uncontrolled study. And if they got something pretty close to that, in a population where the control arm declines at the rate that one thinks, because let's remember, like, patients don't do great on the standard of care. So that even, regardless whether they compare against placebo or compare against standard of care, both of those are going to have negative slopes. And if they have anything more horizontal and can detect it, absolutely. I mean, I think that there's certainly, you could imagine a scenario in which it's approvable based on the phase 2b. I just wouldn't go into it counting on that necessarily, because there's just so much unknowns, again, because that, it's really, to me, this is really just about patient selection, because we don't really know how to think about who. In fact, I was thinking about it a little bit more recently. Just think about it. In today's, so they have a study where you couldn't be on, I think you had to be treatment naive. So you couldn't have been on, in their phase 1 study, you couldn't have previously been on any of the approved drugs. So I guess I was just sort of thinking about that. If I were a physician, treating patients, who am I even going to recommend this trial to, right? Are these patients who are pretty symptomatic and are in pretty bad shape? Or are these patients who are maybe got detected super early and are having already a very benign clinical course and are not very symptomatic at all? I think that I could make a better argument that I'd be offering this to the more mild patients joining that trial, as opposed to the more severe patients. For the more severe patients, I'm basically saying, look, I have a drug that I know isn't great, but it will do something, but it's been shown to do something. Or I have this other thing where I have no freaking idea. Which would you like? And, I think most people, especially if they were symptomatic, might opt for the surer thing, right? Whereas if I'm earlier on, I might say, okay, like, I don't think I'm going to get that horrible. Sure, there's a little bit of risk of being in a clinical trial, but even if this drug doesn't do much, I think I'm probably going to be okay for the next 18 months or so. And then if I'm worse in 18 months, of course, I can go on a pirfenidone or whatever afterwards. So I could imagine that's how I see the bias running in the trial in the single arm trial, that if anything, those patients were probably on the healthier side, if I had to bet. And that makes me concerned that if you did that in a real RCT, either the control arm also is going to be more horizontal than you think, Or they're going to design that to select for patients who are a little bit more advanced, in which case the drug-treated arm will be a little bit more negative slope than you would have expected. Again, doesn't mean that it's not real. Doesn't mean they won't see a difference, but I think that's the big issue here is going to come down to patient selection.
Yeah, I think that was really also the strategy of the company that does probably introduce that bias, as you mentioned, and it was also the reason why the company wanted to do an open label single arm to actually be able to recruit patients, because if it's then placebo controlled, then it will be even tougher to get patients in. And I guess then if the readout, if you would have had this call before the readout, it would have been maybe a disappointment if the data would have been just stabilization, because then we could say for real that these patients were probably just healthier. Considering now the very strong increase, there is probably some subtraction we could expect. Now I'm hypothesizing.
The error bars are pretty big on the those later time points are based on small numbers of patients. And go back and you look at the figure, even comparing to the historical data, the curves don't really start to separate until about 24 weeks. So, and then after 24 weeks, they only had like 9 evaluable patients, I think, at those later time points. So, and 7 even further out. So I'm not sure that I would necessarily be viewing it like that. I think, I was a little disappointed, honestly, that they did not provide more details on really the clinical characteristics of the patients in the single arm trial. Yeah. And I think that, if I were an investor, I'd be pushing for that. And certainly I'd be keeping my eyes very wide open, looking for those data to be disclosed. And I think that once they are disclosed, I'd be really trying to get a sense on how, relatively speaking, do I think those patients were super duper healthy, super duper sick, or somewhere in the middle?
That's a good point, and one you make in the book as well, that one should look for the baseline characteristics that are important and not mentioned by the company. Yeah, so that I fully agree.
Yeah, and I haven't done a search, but at least when they did their call, they kind of, they downplayed that. They said, yeah, they were typical and they look similar to the patients who had participated in other trials. But, the details are important in that case. Again, because we know that this is a disease where there's a population of patients who might be stable for a relatively longer period of time and then enter more of a phase where they, where they're declining more rapidly. So, I think that's really at the crux of it for these guys.
Yeah. Thank you very much. That is very informative. And now I think it's even complex with Vicore, but I think BioInvent is an even more complex case. So the immuno-oncology company has now 4 drugs in clinics. The 4th is about to dose the first patient. Data is out from the lead asset in Non-Hodgkin's lymphoma and a bit of a few patients in solid tumors. How should we look at early trials in immuno-oncology? How can we grade them? How can we see if that is? Yeah,
it's funny. So I've had this conversation with so many clients where I've said, okay, you're going to run this single arm study. We'll talk about the monotherapy combination issue in a second. I'll say, okay, you're going to run your study on, and even without seeing the data, let's just imagine what the data are going to be. You treated 30 patients, you got 1 CR and 3 PRs. Okay, what are you going to do? What is that going to tell you? How are you going to decide what to do next? And I think it's complicated because the answer is, nobody really knows. I think in this case, this is sort of a special case of this, and again, of this problem, and specifically related to TIGIT, which we talked about earlier, right? Yeah. I mean, this is a situation in which there was really as far as I know, there was no meaningful monotherapy there's never been a demonstration of meaningful monotherapy activity. So they've gone straight into a combination study. And, I think 7, 8, 9 years ago, there was this idea that if you did treated these relapse refractory patients or so-called second-line patients, whether you're talking about the heme space or whether you're talking about the immuno-oncology drugs, I think there was this idea that, of course, if you got responses there, that it was all going to be due to the fact that your drug had potentiated the the baseline therapy, because of course, if you just would have come in with the baseline therapy, you wouldn't have gotten any responses, because these patients by definition have quote unquote failed or progressed through first line therapy. I think now we know that that's just not true. And the way we know that's not true is because there have been multiple legitimate RCTs done comparing, for example, PD-1 versus PD-1 plus agent X. That have not shown any meaningful difference, even though the single arm combination study did show some sort of overall response rate, which looked intriguing. So, again, we could have, I guess one could have a discussion about the science behind that, et cetera. But, I think just operationally what it means is that these single arm combination study results in the absence of monotherapy data are just super tough to interpret. And I don't want to be a nihilist and say none of it matters. But on the other hand, I don't think anyone has really great ideas for understanding sort of when to take those seriously and when not to. And a lot of investors and a lot of companies have gotten burned on these types of data. The things that you might want to see in this type of scenario, which we don't see here, unfortunately, would be, first of all, some sort of plausible patient selection hypothesis that you'd imagine would enrich for responders to the combination over just coming back with the baseline monotherapy, with the backbone rather. The other thing that certainly in big companies often gets done, but I think unfortunately doesn't get done as much by small companies just because it's logistically hard and expensive, is to actually run a small trial where of the monotherapy, where even if you don't get responses, you can do post-treatment biopsies and show some sort of legitimate in vivo human proof of mechanism. That's the kind of thing that I think gives people comfort that, okay, at least the drug is doing something meaningful in the body. And then the third thing, of course, is showing some sort of monotherapy activity, which I think puts it in a whole different weight class, so to speak, in these combination sort of setups. So I don't really know what to think. I mean, as you said, there's a lot of these sorts of situations, and they're all complicated. I think
one question here would be then, because it's so complex, could we look at the duration of a response? Is that maybe if the complete responses, if they are long enduring, is that maybe a clearer sign that there could be something, or is it still very hard to judge?
No, it's a fair point. And, I do think that in addition to that, when I've talked to large pharma people about how they evaluate these data, either if it's their own data or they're doing diligence, they look very carefully at not just the length of the responses, but they look really at the clinical characteristics of the particular patients who experienced the response. How heavily pretreated were they beforehand, for example? Not just having received monotherapy, but if you have someone who's sort of 5th, 6th, 7th line, and then gets a complete response, maybe you're more impressed by that than someone where literally this is their second line of therapy that they've gotten, right? So one can do these things, yes. I think from an external investor perspective, we often don't have enough information. The duration idea that you mentioned is certainly interesting. I guess my question there is, I'd be curious to then go to some of these failed RCTs of combinations and see whether there's any meaningful difference. Do you get sort of equivalent when you re-challenge these people with the backbone? Do you still get sort of equivalent duration? And also, what were the or was there really some different? What did the durations look like even in their single arm trials? I think that would be a great little research project, actually. Going back historically and trying to get a sense of whether sort of duration of durability of response, so to speak.
So I guess then I'm sorry.
Yeah, no.
Then if you zoom out from that, then we maybe just conclude that immuno-oncology is very difficult to interpret early. That then maybe from a more investor perspective, one should look that the safety is there, that at least that is not going to be a problem. Maybe the the diversification of a pipeline, that there are some other assets as well. And maybe the kind of herd behavior that we saw in TIGIT, where many specialists invested and pharma did a big deal like last year, GSK, Iteos, that maybe their valuations are already so high that you have very little upside potential left and a big downside potential. And then maybe the smaller ones that are going for not much different targets show similar early data, or then maybe at least more promising in terms of risk reward potential.
Yeah.
Or how do you see that?
Yeah, I mean, I guess I see it similarly. I mean, I remember when people were all jumping on the IDO bandwagon, and everyone would put up a slide with a pretty picture of signaling, and with arrows, sort of like what you'd see in Cell or Nature. And everyone had made these beautiful stories for why the logic was so great that this was going to work, and et cetera, et cetera. And I think I haven't kept up as much with TIGIT, but I assume that it was sort of a similar story, that there was a little bit of a scientific bandwagon jumping where people said, okay, look, we don't have data to support it, but on the other hand, we just sort of fell in love with the story, essentially. So there may be other cases where the mainstream consensus has not fallen in love with the story as much, but maybe the actual strength of the story isn't that much worse. It may not be better, but is it any worse than the story for TIGIT or IDO?
Yeah.
No, maybe not, right? In which case, I think you would say, look, I'm not gonna claim that this is destined to be a home run, but maybe if I invest in 10 of these, maybe 1 or 2 of them is going to work. That's certainly a reasonable strategy, right? I think the negative, the more pessimistic view there would be that so far we do not yet have evidence. I do not believe that we have great evidence for the overall idea that something cannot have monotherapy activity but can robustly synergize with one of these backbone agents. Even though in theory, everyone, that it certainly makes sense that you shouldn't need to have monotherapy activity in order to generate synergy value from clinically. But in practice, I do not, maybe someone will write into you and say that and give examples of where I'm wrong. But I certainly I'm having trouble thinking of an example where that's been shown not to be the case. In other words, if you don't have monotherapy activity, the odds that I think there's probably an evolving consensus that the odds of success of these types of studies is lower than we had thought. All other things being equal. Sorry to be a little bit more pessimistic maybe than one should be, but
I think it reflects a bit the current market sentiment as well, because there were a lot of failures. Alzheimer didn't have the best story now over the last year. Maybe BioArctic will change it. We will see. But so it's a tough field dealing with human biology. And maybe to conclude a bit for investors, we see also now your view that there is, of course, everywhere risk remaining, that at least the valuation should be a bit in line with the risk. And we saw many companies that were bloated up into the billions of dollars with just very early data, sometimes even just preclinical data that also happen, sometimes early clinical data that maybe there one should be a bit more critical and see that these valuations are maybe still just in the hundreds of millions of dollars, and else there is maybe a bit of a bubble risk. And I think mostly deflated now, but I think I still see some companies that decreased a lot. But I would say, as you said in the very beginning, if their asset is not working, then even if they went down from 3 billion to 800 million, it's still too much.
It might be. I mean, I think the other thing that I would say about most of these companies is, what I tend to do in these situations is really just to try to imagine what a plausible path to the market looks like if the company had to run solo. Because I do, again, not to be too negative, but I think that betting that these large companies with a lot of cash on hand are going to come and scoop up phase 1, phase 2 assets, I just do not think that, from my experience, seeing this more from the big pharma side, on the BD side, I just do not think that that is the right bet to make. I think some of these companies will be massive, the ones that can make it all the way to full de-risking will be worth a lot, and they will be bought at huge premiums because there's nothing pharma likes more than revenue. So buying revenue at a premium, no investor gets mad at you for buying revenue at a premium in this business. Whereas buying pipeline with a lot of risk associated with it, doesn't help anybody today, and also potentially doesn't help anybody tomorrow either. So, I think I'd be looking, in terms of, from an investing standpoint, I'd be looking for companies and assets where there's a plausible way that you could imagine getting all the way to the end, and maybe shying away from scenarios in which the company really seems like it's going to basically need some sort of partnership or an acquisition for the asset to move past a certain point.
Yeah, I think that is very true, and maybe we take that as the final statement. So companies should be able to go all the way themselves, and we should not wait for pharma to jump in before, while it can always happen, of course.
It can always happen. And again, there's also, I guess also from an investor, the last thing I would say there is, to remember that from an investor point of view, a lot of people get excited about M&A, but when you're actually in the biotech, a really meaningful licensing deal, with some staged payment of cash, can be super valuable in terms of kind of continuing the process. And I think that's more likely. So, if you're patient, again, so that's a scenario in which you think that the clinical development plan is set up to really get to those meaningful inflection points where the company could credibly go to Pfizer or Merck or whomever and say, look, we've taken a lot of risk out of the equation. That's why, when you look at some of the examples that we saw, like in the IPF case, for example, certainly those initial data are intriguing. I think this upcoming study is probably going to be for the large companies that are looking at this space. I think the upcoming RCT is going to be a big driver of what they view as the overall risk and value of the asset. And if there's, if it seems to be acting as advertised, do I think they would get acquired right after that? Again, I'm pessimistic about that. Do I think they would have a chance at a meaningful partnership after that? I would think so. Again, assuming the data looks something like what they showed in their single-arm study.
Yeah. Thank you very much, Frank, for your time. That was very insightful. Sure our viewers will agree. And thank you everyone for watching.